Hey buddies, in this video I'll talk about tomato. The scientific name of tomato is Lycopersicum esculentum, belongs to family Solanaceae and chromosome number is 24 in diploid form. Tomatoes are rich source of vitamin A. These are exceptionally rich source of lycopene. Tomatoes may have 346 and 785 microgram beta and alpha carotene respectively and 23 milligram per 100 gram of vitamin C. Tomatoes have antioxidant activity amongst free radicals responsible for many actual important diseases like cancer. One single improved tomato can provide a person's full daily vitamin A requirement. Types of tomato. They come in different forms, shape, size and color. The preference is based on fresh market, processing and cherry type. Progress of wild tomato introgression. There are different wild sources of tomato which contributed for quality and quantity improvement as well as biotic and abiotic stress. Lycopersicum hirsutum contributed in order to avoid fruitworm, CMV and PVY. And the improved one is Lycopersicum esculentum. The other source is Lycopersicum esculentum serraceae that contributed genes for a resistance for fruitworm and bacterial wilt. Likewise, Lycopersicum pennelli for fruitworm, Solanum pimpilifolium for late blight, Lycopersicum peruvianum for root knot nematode and taspovirus, then Lycopersicum chilens for TLCV. This is the picture showing diversity in tomato or Lycopersicum species. They are available in different form. The green tomatoes are consumed for different purpose, red are consumed for different purpose, and the cherry type are specifically used for sambar preparation. This is the scenario of tomato production in India. The rank about production area and productivity. India rank 4th in production, 2nd in area and 98th in productivity. There is often a question arise in mind whether tomato is a fruit or vegetable. The answer is even though it is consumed in the ripened stage, it is considered as a vegetable. Importance of tomato. It's the second most consumed vegetable after potato in the world. First in terms of total contribution of nutrients to human diet among vegetables. It is consumed in fresh form and various processed forms like soup, sauce, ketchup, paste, puree, canned, etc. It tops the list of processed vegetables, also an ideal model plant for physiological, biochemical, cytological and molecular investigations. Coming to origin and evolution of tomato, it originated in New World, that is South America, especially to the Andean region encompassed by parts of Chile, Colombia, Ecuador, Bolivia and Peru. It was domesticated in Mexico by Indians. Immediate ancestors of Solanum Lycopersicum esculentum are Lycopersicum esculentum variety serraciforme, that is wild cherry tomato. After Spanish conquest of South America spread to Europe, USA and rest of the world. Concomitant gradual evolution of the large fruited type and shift from allogamy to autogamy under domestication. Coming to different species, Lycopersicum esculentum, Lycopersicum pimpinellifolium, Lycopersicum cheesemani, hirsutum, pennelli, milevsky, then parviflorum, chilens and peruvianum. Inflorescence of tomato. This has extra axillary cyme with dichotomous or polycotomous branching. Here the branching habit is given. The flower is abracteate or ebracteate, bright yellow, chasmogamous, pentamerous, actinomorphic, hypogynous, hermaphrodite with petals enveloped by a solid tube formed by the stamens. Calyx. The sepals are 5 united, often persistent, and possesses trichomes. Corolla is bright yellow with 5 petals and is gamopetalous. Coming to androecium, it consists 5 stamens, epipetalous, small filaments, large anther. Stamens form a solid cone enlarging the enclosing the pistil. Then anther dehiscence is introsely longitudinal. The gynoecium part is consisting of stigma, style, ovary. The ovary is superior, bicarpellary and syncarpous. Style is single and stigma is bilobed. This is a dissected flower and the labeled different parts. This is morphological description of flower. Coming to anthesis. The anthesis starts in the morning around 6 am and continues till 11 am. Maximum flower opening is from 7 to 9 am. Stigma receptivity is for 16 to 18 hours before anthesis to 5 to 6 days after anthesis. Pollen viability remain for 2 to 5 days at 18 to 25 degrees Celsius and up to 6 months in the desiccator, that means under cold temperature at 5 degrees Celsius. Coming to pollination, essentially tomato is a self-pollinated crop as the stamens form a solid cone enclosing the pistil. Self-pollination varies between 94 to 99%, but 1.5 to 47% natural cross-pollination have been reported depending upon temperature, insect population and activity at flowering time, planting design, plant population, wind velocity and direction, then genotypic nature. Fertilization and fruit set. The maximum fertilization and fruit set if pollination is at the time of anthesis. Fertilization occurs 24 to 54 hours after pollination. It takes 6 to 8 weeks from the first flower to ripe the fruits. Fruit is a berry. Coming to selfing techniques, bagging of young buds, then manual transfer of freshly collected pollen on stigma of flowers of the same plant. Crossing techniques include emasculation, pollen collection, and artificial pollination. Selection of flower bud. To select a flower bud in which anthesis will commence 12 to 16 hours later. Petals just out of the bud and corolla color should be slight yellow or even paler. For emasculation, remove the male organs from flower of female parent. Use sharp pointed forceps to force open the selected buds. Then split open the anther cone. Grasp the anther cone with forceps with one tip inserted between style and anther and the other between anther and calyx. Gently pulling away from the floral axis, carefully remove the anther cone out of the bud, leaving the calyx, corolla and pistil. Or the stamens can also be removed individually. Bag the emasculated flower. Then normally emasculation is done in the afternoon and pollination in the next morning. To avoid emasculation, CGMS or GMS lines can be used in hybrid seed production. The pollen collection process. Collect flowers from male parent during early morning hours. Remove the anther cones from the flower and put in a glass envelope. Pollen collection. For this, dry the anthers by placing the envelopes 30 cm below a 100-watt lamp for 24 hours. The temperature is around 30 degrees Celsius. Put the dried anther cones in a plastic cup. Cover the cup with a fine mesh screen of 200 to 300 mesh and then seal it with a similar tight fitting cup, serving as a lid. Shake the cup about 10 to 20 times so that the pollen is collected in the lid cap. Transfer the pollen into a small, suitable container for pollination. Fresh pollen is best for good fruit set. It can be kept for 1 day at moderate room temperature or can be placed in dehydrator with silica gel and kept in a refrigerator at 10 degrees Celsius to maintain viability for 10 to 15 days. For small quantity of pollen, we can use or vibrate the flower to release the pollen and collect it in a glass tube, or use a flattened dissecting needles or forceps to gather pollen by cutting a slit in the anther cone and scraping the contents of one antheridia at a time. Artificial pollination. In this, one complete blossoming of the emasculated flower, cut their calyx and corolla to expose the stigma. Dip the stigma into a pool of pollen in the pollen container or pollinate by touching the stigma with the tip of index finger, dipping in the pollen pool. Freshly collected pollen can also be applied with a camel hairbrush or by using a pollination syringe. Then hybridized flowers are tagged properly. Pollination is usually done 3 times weekly on alternate day over 3 to 5 week period depending upon the rate of flower development. Successful pollination results in ovary enlargement within 4 to 5 days as seen in the picture. Success rate is 50 to 70%. It depends upon temperature and humidity. 22 to 28 degrees Celsius and 70 to 80% relative humidity are optimum for good seed set. Pollinate only first 3 to 4 flowers in each inflorescence, as basal flowering set seeds better than terminal ones. Coming to hybrid fruit production, this can be recognized by the cut sepals. 2 methods of hybrid seed production are hand emasculation and pollination, and second is use of male sterile lines. First, emasculation. For this, emasculation has to be carried out after 55 to 65 days after sowing. Then flower buds which will open in 2 to 3 days are selected and the petals will be slightly out of the flower bud but not opened. Like this we have to remove. This has all discussed already, but I am summarizing it once again because the method of pollen collection and pollination is entirely different in tomato as compared to other crops. Coming to heterosis in tomato, the history of tomato heterosis. The phenomena was first noticed by Hedrick and Booth in 1908 for yield and fruit. In 1946, first hybrid tomato cultivar single cross was released by Dorst. Breeding goals in tomato has gone through 4 phases: for yield during 1970s, for shelf life during 1980s, for taste in 1990s, and for nutritional quality it is going on. Gilbert in 1912 reported 300% increase in total yield through heterosis. In India, tomato improvement was initiated during 1950. In 1973, IAHS released first tomato hybrid, Karnataka. Coming to breeding objectives in tomato, early and high yielding varieties or hybrids, resistant to pest and disease, resistant to root knot nematode, suited for cold and high temperature, suited to prolonged storage and transport, suited for processing, then for multiple utility as well as multiple insect and disease resistance. The next one is for quality improvement like nutrients, minerals and all those things. Different developed tomato hybrids are enlisted here and their peculiar characters are also given in public and private sector. First one is Arka Vardhan, Arka Abhijit. All Arka series are developed by IIHR Bengaluru. Arka Sreshtha, Arka Vishal, Arka Ananya. Then from IRI New Delhi, Pusa Hybrid 1, Pusa Hybrid 4. Private sector hybrids, Rupali, Naveen. From Mahiko Company, MTH4, Gulmohar, Sadabahar. Then IAHS Bengaluru developed Rashmi, Vaishali. Then Novartis seeds, Avinash 2, Sonali. Namdhari seeds developed NS 566, NS 1359. Coming to germplasm collection, Tomato Genetic Stock Center is located at California. Asian Vegetable Research and Development Center, Taiwan. Then IPGRI, IBPGR, New Delhi. Indian Institute of Vegetable Research, Varanasi. Future trust in tomato, development of varieties or hybrids suitable for fresh market and processing, development of cherry type of tomato varieties and hybrids, to breed for multiple disease resistance, to develop varieties for biotic and abiotic stress combinations. Thank you. Hope you liked this video. If you liked, don't forget to subscribe my channel. Thanks once again.